So again, the monocytes primarily are going to be in the blood. The macrophages primarily are going to be tissue specific. So when the monocytes migrate to a tissue right through the bloodstream, they'll eventually go on to form a macrophage. And so it's for this reason that you're not really going to see macrophages in the bloodstream. It's kind of similar to what we talked about with mast cells, right? Mast cells, you're not really going to see them in the bloodstream because they're going to essentially get activated at the tissues, right? All right, going through the structure of these macrophages. So there's going to be, specific receptors. One of the big ones are these FC receptors. Remember, macrophages, this makes sense if we think about what their primary function is, right? The big function here is going to be phagocytosis, right? We're going to be phagocytosing invading pathogens, similar to what we saw with neutrophils. The big difference here is that the macrophages are essentially leveraging this phagocytosis to eventually go on to form antigen presentation. Okay, so antigen presentation is what's going to be done through their MHC2 receptor. And all of this is, discussed in great detail in the immunology unit. So I'm not going to go through all the details here, but essentially you're going to take in these pathogens, you're going to break them down, and then you're going to present their antigens on MHC2, right? You're going to use that MHC2 receptor to bind to a CD4 positive T cell. Okay, so you're going to bind to a CD4 positive T cell using that MHC2 receptor, right? Binding to the CD4 positive T cell receptor. And then there's a co-stimulatory signal, B7-CD28, which, like I said, we discussed in the immunology section. And this activation of this CD4 positive T cell is going to occur through the release of interleukin 12. And so just to recap this, right? If we had some pathogen here in the body and the macrophage goes on to phagocytose this pathogen, macrophage can essentially present components of this pathogen to this CD4 positive T cell. And the idea here is that we're trying to generate a sustained memory response to go down one of these specific T helper pathways. Okay, so the MHC2 is going to bind to the CD4 positive T cell receptor, right? Your rule of eights. This CD4 positive T cell will then get stimulated through this binding as well as this B7-CD28 co-stimulatory signal. Again, all of this discussed in the immunology section. Okay, so this cell is going to get activated. And when it gets activated, there's also going to be some interleukin 12 released from this macrophage. And this interleukin 12 will help stimulate this T cell into a very specific T helper 1 cell. And then that T helper 1 cell will then go on to release interferon gamma to then further activate this macrophage. So with all of that being said, right, this kind of all goes hand in hand. This macrophage undergoes phagocytosis and then presents those antigens via MHC2. Now for the macrophage to undergo phagocytosis, sometimes we might opsonize this bacteria. So we might put things on this bacteria or this pathogen so that the macrophage knows to come over and phagocytose it. One of those things that we can do is we can put IgG on that bacteria, or we can use from our complement cascade C3b, right? Your two big opsonins are going to be C3b and IgG. Let's just say this was IgG. The Fab portion would have bound to the pathogen, and the FC portion would have been the site that the macrophage binds to. So that's why we use FC receptors to bind to the FC portion of IgG, right, when we want to phagocytose a pathogen, and we have complement receptors to bind to things like C3b that can also mark a pathogen for opsonization, right? So this helps the opsonization process and gets us to eventually phagocytose that invading pathogen, which then eventually will lead to antigen presentation, right, via the MHC2 receptor, which is essentially this whole process. And like I said, this process is also discussed in the immunology section if you want a clean look at it. Now, the other thing on these macrophages are these pattern recognition receptors. These are very non-specific receptors that will bind to things like lipid A from lipopolysaccharide. That's probably the highest yield one to remember. And there's a lot of different bacteria that have lipopolysaccharide and have lipid A, right? And these receptors are just going to recognize this as like an early kind of surveillance signal that something is wrong. There's lipopolysaccharide in the body that's not supposed to be there. So this lipid A will essentially bind to these CD14 toll-like 4 receptors. And there's variants of this. This is probably the highest yield one to remember. And again, this is a non-specific receptor that just says, hey, there's lipid A here. It's not supposed to be here. We need to set off an acute inflammatory reaction. And so this reaction is set off releasing things like interleukin 1, right? TNF-alpha, for example. These substances are released in response to this non-specific lipid A that's in the body from this bacteria, right? That's usually coming from lipopolysaccharide. And so we can see all of that here. So cytokine production, you'll see from macrophages, right? Interleukin 1, part of the acute inflammatory response, right? Causing you to have things like a fever. TNF is another classic one that's released. And then interleukin 12, this is going to be more consistent with stimulating this T helper 1 response that we saw down here. Okay, so not so much associated with the acute inflammatory cascade, more so associated with stimulating a specific type of T helper 1 response. And we see this very frequently, this T helper 1 response, in the setting of granuloma formation. Okay, so when macrophages are essentially walling off an invader or something they feel like they can't digest. like an intracellular bacteria, for example, mycobacteria and tuberculosis is very classic for this, right? So we get granulomas here, classically caseating granulomas that have necrosis. And then also we can see this in sarcoidosis being more of the non-caseating variant, okay, of the granuloma. And so you can see an image here. This is an example of a granuloma. You can see this pretty dense collection of leukocytes. Okay, so you have a lot of inflammatory cells kind of out here on the outside. Here you can see a little bit of necrosis. You can see these white kind of spaces in here representing destruction of this tissue. There's not a lot of cells, right? This is relatively acellular in this region. So you can see a little bit of necrosis here, signifying that this is probably more of a caseating type of granuloma, more associated with TB. And then the last thing, the important thing here, is that, when a situation where these macrophages are having a tough time digesting certain organisms or structures, foreign bodies in general, sometimes they'll fuse to form these giant cells. Okay, and we talk about all of this in much greater detail in the immunology section, but essentially we're forming these giant cells to try to become more capable of essentially engulfing, this bacteria. And so you can see this is an example, very classic example here. And the last thing here, when we're talking about macrophages, like I said, there's some variance to this, but macrophages are going to be tissue specific. And so if we go by tissue, just some cells that you probably have run into at some point, just remember that essentially these are all macrophages, but they're slightly different in some of their characteristics and things that they do. And so they have some unique names. So the Langerhans cells are very classically seen in the skin, the Kupffer cells in the liver, microglia, we can see that in the brain, the spinal cord, and then the osteoclasts, very classic in bone tissue.